Okay, so welcome to this next video in which we are discussing gastric acid secretion. Okay, so we've now discussed the mechanism by which parietal cells secrete hydrochloric acid from the basolateral side onto the apical side. Okay, so how they secrete hydrochloric acid into their canaliculi, which then goes into the lumen of the stomach. Okay, so we now want to discuss the factors controlling the secretion of hydrochloric acid by these parietal cells. So basically, nearby the parietal cells, if I just draw another one of these parietal cells, okay, so here is the basement membrane, okay, and then we have our parietal cells sitting on the basement membrane here, like so. Then basically, we know that underneath the basement membrane is this layer that we've called the lamina propria. So this layer that I've highlighted in yellow. Basically, in that lamina propria, there is a special type of cell whose job is to control the rate of secretion of these parietal cells. Okay, so let me put this cell here. And they sit extremely close by the parietal cells. So this cell is called an enterochromaffin-like cell, okay? Because they are similar to the enterochromaffin cells within the small intestine, the wall of the small intestine. Okay, so this is an enterochromaffin-like cell. And for short, enterochromaffin-like cells are often abbreviated to E for entero, C for chromaffin, L for like, so ECL cells. Okay, now, basically, these ECL cells secrete histamine, okay? So they secrete histamine, and this histamine will diffuse over to the parietal cells and stimulates these parietal cells to secrete hydrochloric acid. Okay, so this is a form of paracrine signaling. So when one cell secretes a molecule which then diffuses to the surrounding cells and causes some sort of activity in them, that's known as paracrine signaling. Okay, so let me show you the structure of histamine, and then we'll discuss the histamine receptor that is on the surface of the parietal cells which the histamine acts on. Okay, so basically, the structure then of histamine, we'll draw it down here. Okay, so histamine is based on the amino acid histidine, but it's had the carboxylic acid group removed. So let's try and draw it in parallels. So here's the amino group, here's the alpha carbon of the amino acid. Now there would be a carboxylic acid down here if we were drawing the amino acid histidine, but we're not, we're drawing histamine, so instead there's a hydrogen there, and then the R group of histidine, which is the same in histamine, is then a methylene group with an imidazole ring here. Now an imidazole ring is basically a 5-membered ring where 2 of the members are nitrogens and 3 of the members are carbons. Okay, and you have these 2 double bonds. So this ring that I'm drawing here is called an imidazole ring. Okay, and basically the reason it's called an imidazole ring is that it's got this special double bond here between the carbon and the nitrogen. Now whenever you have a double bond between a carbon atom and a nitrogen atom, it's known as an imide bond. Okay, so that's where the imide of the imidazole ring naming system comes from. Okay, the dazole makes sense because azo means pertaining to nitrogen, and dazole kind of implies that you have 2 nitrogens. So imidazole is kind of a nice name for this. Okay, and now we just saturate everything else by putting on hydrogens. Okay, so this is the structure of histamine. If you wanted the amino acid histidine, you'd have to take this hydrogen off and put a carboxylic acid group there instead to turn it back into an amino acid. So basically, these enterochromaffin cells here secrete histamine at a basal level. Okay, so they're always secreting a little bit of histamine, and this goes over and acts on special receptors on the surface of the parietal cell called H2 receptors, which is a form of G protein coupled receptor, which we'll discuss more in a moment. Okay, and this histamine causes the parietal cell to continue to release gastric acid, basically. So there is a basal secretion of histamine always from these enterochromaffin-like cells, which tells the parietal cells, keep going, keep releasing a basal level of hydrochloric acid. Okay, and the way that this occurs is because the histamine acting on the H2 receptors is overall going to result in the activation of these proton potassium ATPases, these proton pumps basically, which after all are the thing which drive the entire secretion of hydrochloric acid. So I now want to look at the pathway by which histamine acting on H2 receptors actually leads to the activation of the proton potassium ATPase. Okay, so basically it's a GS pathway. So let's start this process off then. So let's say this is the phospholipid bilayer of the parietal/oxyntic cell. So here is the parietal/oxyntic cell. And in its phospholipid bilayer, it has receptors for histamine. And these receptors are of the H2 type. Okay, so here is the H2 receptor. Now it is a G protein coupled receptor. Specifically, it's a G protein coupled receptor within the family of rhodopsin-like G protein coupled receptors. So basically, G protein coupled receptors can be classified into 5 separate families. The rhodopsin-like family of G protein coupled receptors, which is the biggest family of G protein coupled receptors, it contains 750 different members. Okay, and that's out of the around 800 members of the G protein coupled receptor family. So it contains most of the G protein coupled receptors. But then there's also the secretin G protein coupled receptor family. There's the glutamate G protein coupled receptor family. There's the adhesion G protein coupled receptor family. And then finally, there's also a sillily named G protein coupled receptor family, which is the frizzled taste2, frizzled / taste2 G protein coupled receptor family. Okay, named after the 2 main G protein coupled receptors that are within it, which are the frizzled receptor, which is involved in the Wnt beta-catenin pathway, and the taste2 receptor, which is involved in gustation. Right, okay, so H2 is a G protein coupled receptor, so it has this characteristic 7 transmembrane domain structure, and it's within this rhodopsin family of G protein coupled receptors. Now, what does that actually mean? It means that the ligand binds to the G protein coupled receptor by binding to residues that are within the transmembrane domains. So basically, histamine will bind to residues within the transmembrane domains, these membrane-spanning alpha helices. That's the case for all rhodopsin family G protein coupled receptors, that their ligands bind to them by interacting with residues in the membrane-spanning alpha helices. Okay, so when histamine comes in, it will interact with residues in the membrane-spanning alpha helices, and what it will trigger is a conformational change in this histamine 2 receptor, such that the histamine 2 receptor can now interact with a heterotrimeric G protein. Now, the change will occur on these 3 intracellular loops. Okay, so this one, and this one. So you have these 3 intracellular loops. The first intracellular loop, called ICL1 for intracellular loop 1, is between the first and the second membrane-spanning alpha helice. The second intracellular loop, ICL2, is between the third and the fourth membrane-spanning alpha helice, and the third intracellular loop is between the 5th and the 6th membrane-spanning alpha helixes. Okay, so, right, basically, when the ligand binds, when histamine binds to the transmembrane domains of the H2 receptor, what will happen is you'll get a conformational change in the intracellular loops here, so that there is now a binding site for a heterotrimeric G protein. Now, which heterotrimeric G protein is going to bind here? Well, it's going to be a heterotrimeric G protein of the GS archetype. Now, what does that mean? That means that basically it is a heterotrimeric G protein where the alpha subunit is an alpha S subunit. Okay, so the alpha subunit is going to be the G alpha S protein, but it does not tell you what the beta and the gamma subunits are. So let me just show this. So basically, heterotrimeric G proteins consist of an alpha subunit, a beta subunit, and a gamma subunit. Okay, so here is the alpha subunit. Now, basically, like all G proteins, and there are more G proteins than just the heterotrimeric G proteins. There are also the monomeric G proteins, of which RAS would be the archetypal example. Okay, now all G proteins have two states. They have an on state and an off state. They are effectively switches. Now when they are in the on state, they have GTP bound to them, and when they are in the off state, they have GDP bound to them. Okay, so let's say that originally our alpha subunit is in the off state and therefore has GDP, guanosine diphosphate, bound to it. Okay, now the alpha subunit is attached to the inner leaflet of the phospholipid bilayer via a lipid modification. Basically, it's had a lipid group, a long-chain carboxylic acid group attached to it, and this anchors into the inner leaflet of the phospholipid bilayer and holds this alpha subunit at the inside of the lipid bilayer, basically. Okay, so that's how they remain attached to the inner leaflet of the phospholipid bilayer. Now, when alpha subunits are in their off state, basically they can bind now to another complex of proteins. Okay, so they can bind to what is known as the beta-gamma complex. Okay, so basically there are two other proteins that make up a heterotrimeric G protein. So let me just write the name out. We're talking about heterotrimeric G proteins. Okay, so these proteins are made up of 3 separate subunits. That's what the trimeric tells us here. And these are 3 different subunits. That's what the hetero tells us. Okay, so basically heterotrimeric G proteins consist of an alpha subunit, a beta subunit, and a gamma subunit. Okay, now the beta and the gamma subunit always remain bound together, and it's a good job because the gamma subunit has a lipid molecule, again, that's been attached onto it and which anchors into the inner leaflet of the phospholipid bilayer and holds the beta-gamma complex, as it's called, to the inner leaflet of the phospholipid bilayer. But the beta subunit has no such lipid modification, so if it weren't for the fact that it holds on to the gamma subunit, it wouldn't stay bound to the inner leaflet of the phospholipid bilayer. Now, there are a huge number of different alpha subunits, there are a huge number of different beta subunits, and there are a huge number of different gamma subunits. In fact, there are 16 different genes for alpha subunits, and then 21 actual different subunits, which occurs because there are different splice variants of each gene. Okay, there are 5 genes for beta subunits, and that gives rise to 6 different subunits, again because one of the genes has 2 splice variants. And then finally there are 12 genes for gamma subunits, which gives rise to 12 different subunits. That one's nice and simple. Okay, so basically when we say that the heterotrimeric G protein is a GS heterotrimeric G protein, all that tells you about is what the alpha subunit is. That tells you that this alpha subunit is specifically an alpha S subunit. So one of these 16 genes is the G alpha S gene, okay? Now it does have different splice variants, but they all pretty much do the same thing, so we won't dwell on that. We'll just call this an alpha S heterotrimeric subunit of a G protein, an alpha subunit of a heterotrimeric G protein. Okay, so basically we don't know, neither do we care, what the beta and the gamma subunit are. This doesn't tell us that. Okay, So what is known is that the H2 receptor will interact with a heterotrimeric G protein of this GS type, which means that the alpha subunit will be alpha S, but we don't know what the beta and the gamma subunit will be. Okay, right. Now, in the inactive state, where the alpha S subunit has GDP bound, it will associate with the beta and the gamma complex to form a heterotrimeric G protein. When histamine binds to the H2 receptor and changes the conformation of these three intracellular loops, what will happen is the heterotrimer now will bind to this binding site on the cytoplasmic side of the H2 receptor, and what that will trigger is it will trigger the alpha S subunit to lose its GDP, and instead GTP from the cytoplasm will come in and bind to the alpha S subunit instead. Now, as soon as this happens, what happens is the alpha S, now bound to GTP, leaves the clutches of the H2 receptor and goes off. Now, it will still be attached to the inner leaflet of the phospholipid bilayer because it's still got this lipid modification, but it doesn't stay bound to the active H2 receptor. Okay, so here comes the alpha S subunit, now with GTP bound. And it no longer wants to associate with the beta and the gamma complex either. Now, the beta and the gamma complex are also released from the clutches of the H2 receptor, and they go off as well. Okay, now what's going to happen is that both of these structures, the beta gamma complex and the alpha S GTP, can go off and activate downstream targets, but we will focus on the downstream targets activated by the alpha S GTP subunit. Okay, and we'll see these downstream targets in the next video.